Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUET exam question paper for the subject Biochemistry conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Biochemistry being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist C for CUCET, and there you will find. So coming to the first question. In a substrate enzyme reaction, the term Ka denotes which of the following? So it is nothing but your association constant. Then equilibrium constant, it is nothing but the ratio of the concentration of products to the concentration of reactants. Dissociation constant, it is the reciprocal of the association constant. Then turnover rate, that is your Kcat, already discussed so many times. Then a metabolic condition in which the capacity of the body to buffer OH minus ions is diminished. It is called as alkalosis. Okay. Kurtosis, it has something to do with your biostatistics. Acidosis, all that I have discussed earlier. Lipidosis also, it is characterized by accumulation of excess lipids. Then the immune cells that ingest large particles and cells by phagocytosis are called. So those immune cells that ingest by phagocytosis are called macrophages. B lymphocytes, they produce antibodies. Cytotoxic T cells, they produce certain chemical signals and activate other immune responses. Helper T cells also, they assist in activating other immune responses. Then enzymes that are involved in the transfer of electrons are called. They are called oxidoreductases. Okay, they are not transferases, it is transfer of electrons or hydrogen atoms or hydride ions. It is oxidoreductases. Transferases are enzymes that catalyze transfer of functional groups between molecules. Hydrolases, they catalyze hydrolysis of bonds. Ligases, they catalyze formation of new chemical bonds between molecules. Then among the following enzymes, which one has the highest turnover number? It is your catalase. Then albinism is a genetic disorder associated with skin. Albinism, it is affect the production of melanin. There is little pigmentation in the skin, hair, eyes, etc. So here it is skin. It is albinism is associated with the skin. Of the following pH values, which one is the lowest? So on a pH scale, pH 0 is the lowest and it indicates a strongly acidic solution. 14 value indicates strongly basic solution and 7 is for neutral solution. So the answer here is option 1, 0. Which among the following liver function test, which one indicates the hepatocellular damage? It is AST. AST is aspartate transaminase and it is primarily found in the liver cells, that is your hepatocytes. So when these hepatocellular means the hepatocytes, that is liver cells, are damaged. So when these liver cells are damaged, this AST will be released into the bloodstream. Then alkaline, ALP is alkaline phosphatase. So it is also elevated in liver damage but not during hepatocellular damage. BIL is bilirubin. It is elevated in the case of jaundice as well as liver dysfunction. Then gamma glutamyl transferase also elevated in liver disease but does not indicate hepatocellular damage. Then the next one. The cause of increased gamma glutamyl transferase GGT in human serum can be due to. So it can be due to gamma glutamyl transferase. It is an enzyme found in liver, pancreas and kidneys. So due to alcohol consumption, excess alcohol consumption or chronic alcohol consumption can cause liver damage and GGT can increase. Hepatitis also, it can cause increase in GGT levels. Pancreatitis also, it can cause increase in GGT levels. And liver metastasis, that is in liver cancer. Metastasis is a characteristic property of cancer cells. So liver metastasis is cancer, that is, so it can cause increase in GGT level. So A, B, C, D is the answer. So answer is 3. Then human acute liver failure can be due to. It can be due to viral hepatitis, yes. Due to drug induced, that is medicines. It can be due to medicines. It can be due to toxins, yes. It can be due to ischemia, shock or hypertension. So D is also, these are all 4 are correct. So your answer is 3, that is A, B, C, D. Then read the following statements carefully. Ultracentrifugation separate molecule based on their density. Yes, based on their size and density, ultracentrifugation separates. Gel electrophoresis separate molecule based on their mobility in an electric field. That is also correct. Based on their size and charge, using an electric field is gel electrophoresis separate molecules. Then gel filtration separate molecule based on their size. Yes, gel filtration, it is having a porous gel matrix. The smaller molecules, they will enter into the pores of the gel, whereas the larger molecules, they cannot enter into the pores of the gel and they will be eluted in the beginning and the smaller molecule will elute last. So that is also correct. NMR separate molecule based on their proton signal. As per me, that statement also is correct. But in your answer key, it is A, B, C that they have given. But for me, A, B, C, D is also correct. So as per me, 3 is the answer. Then next one. 1 angstrom is equal to. So one thing I will tell you. 1 nanometer is equal to 10 angstroms. Okay. 1 nanometer is equal to 10 angstroms. So 1 angstrom will be equal to how many nanometers? 1 by 10, that is 0.1. So the answer here is option 1. Then in 1953, Frederick Sanger determined the amino acid sequence of which protein? So Frederick Sanger, he determined the amino acid sequence of insulin. Caspase, it is involved in apoptosis. Hemoglobin, you know, oxygen carrier in the red blood cells. DNA polymerase involved in DNA replication. So these amino acid sequence of these 3 enzymes are not determined by Frederick Sanger. It is insulin that he has determined. Then next one. A substance that receives electrons in an oxidation reduction reaction is called. So the substance that receives electrons. So it has to be an electron acceptor. Because oxidation reduction reaction is redox reaction. Oxidation is loss of electrons and reduction is gain of electrons. So electron acceptor, it is a substance that receives electrons during redox reaction and thereby getting reduced. Electron donor is the one which donates electrons in a redox reaction. What is eluate? It is a substance that is eluted from the chromatographic column upon washing. It has nothing to do with oxidation reduction reactions. And electron carrier, it is the one which carries electrons. In a redox reaction, it does not accept or donate electrons. Then next one, the substrate concentration at which an enzyme catalyzed reaction proceeds at 1/2 of its velocity. So it is nothing but your Km value. Km is the substrate concentration at which the reaction proceeds at half its maximum velocity. That is called as Km. CUET, it is the turnover number. I am explaining it since the beginning. They have asked so many questions. CUET was one of the option. V0, it is the initial velocity, and Vmax is the maximum velocity of an enzyme catalyzed reaction. And the maximum velocity, it is obtained when the enzyme is fully saturated with the substrate. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.